Good morning, everyone. We will begin in 1 minute. We're just allowing people to be admitted to the room. So we will begin the webinar in approximately 1 minute. Okay, people are still coming in, but it looks like most of the people are here and we're 1 minute over, so I will get started. On behalf of our program team, I welcome you all to today's webinar about the Research Opportunity Announcement OT 25004, which was issued on March 6th. I am Dr. Matt Arnegard, the Office of Strategic Coordination Program Leader for this initiative, and I'm also serving as the Other Transactions Program Official for this research opportunity announcement. This announcement invites applicants to support the Newborn Screening by Whole Genome Sequencing Initiative, which is a milestone-driven study of the feasibility of a collaborative, multi-state model for incorporating genomic sequencing as a screening tool into the existing state-based U.S. public health newborn screening program. This funding opportunity is a part of the Common Fund Venture Program. Today, we'll briefly introduce the Common Fund Venture Program because, as I just said, this initiative falls within this program. Then we'll introduce the ROA and highlight some of its key points. Lastly, we'll have a question and answer session for the rest of the hour. Please post your questions using the Q&A function highlighted in the red box in the Teams ribbon at the top of your screen. The NIH Working Group will address as many questions as possible during the webinar. If we can't get to all of them, we'll follow up individually with attendees whose questions we didn't answer. And questions and answers that relate to a broader audience will be posted on the frequently asked questions page for this initiative on the Common Fund website, and that web address is given at the bottom of the slide here. We'll post those questions anonymously, so your names won't be included, and we'll also be posting a recording of this webinar, including the Q&A session, for those who could not make it or want to watch it again. So let's start with an introduction to the NIH Common Fund and the Venture Program. NIH Common Fund moves the NIH mission forward faster. It does this by supporting a series of bold scientific programs, each of which is designed to catalyze discovery in an area of research relevant to the NIH mission. These areas of research supported by the Common Fund are cross-cutting rather than focusing on one disease or organ system, and they advance the missions of multiple NIH institutes and centers. Common Fund programs are designed so that each program will spur subsequent biomedical advances that otherwise wouldn't be possible without our strategic investment. The main idea behind the venture program is to do amazing things with modest funding. This new area of Common Fund support provides a framework for the development of short-term initiatives that embrace scientific risk. Venture program initiatives must meet the 5 program criteria listed on the left, which apply to all Common Fund programs. To be a venture initiative, we also expect the initiative to be bold, in other words, to make a daring investment that has potential for significant outsized impact. We want venture initiatives to be nimble so we can implement them rapidly in response to specific scientific opportunities. And these initiatives are focused on clearly defined research topics with a limit of 3 years of funding and up to $5 million of support per year. And now I'm pleased to introduce Dr. Dominique Pichard, who will give an overview of the ROA. Dr. Pichard is the Director of the Division of Rare Diseases Research Innovation at the National Center for Advancing Translational Sciences, or NCATS. She serves as coordinator of the Newborn Screening by Whole Genome Sequencing Working Group and is the primary scientific contact on the ROA. Dr. Pichard, please continue with the presentation.
Thank you very much. Next slide, please. The Newborn Screening by Whole Genome Sequencing Collaboratory, which I'll refer to as NBSxWGS for the remainder of this presentation, aims to demonstrate the feasibility of a collaborative model for broader genomic sequencing across multiple states to demonstrate the potential for a national newborn genetic screening program that maintains the public health goals of identifying severe conditions that are treatable early in life and can keep pace with the therapeutic development landscape for rare diseases. On this slide are listed 4 high-level goals of the program. This initiative has an award budget that is limited to no more than $4.8 million in total costs per year for a maximum of 3 years. No more than 1 meritorious award will be funded. Next slide, please. 97% of newborns in the U.S. are screened by state public health labs, and most of that screening is via biochemical assays on dried blood spots. Many states are able to screen for the 38 core conditions on the federally recommended Uniform Screening Panel, or the RUSP, and we know that thousands of rare genetic diseases are not screened for. This initiative will support a collaborative, multi-state model for newborn screening that would use the whole genome sequencing platform to support a first-tier screening assay for a set of genetic conditions that are actionable in the first year of life. Next slide, please. To carry out the feasibility study, the Newborn Screening by Whole Genome Sequencing Collaboratory will need to consist of the components shown in this figure. Some of the components are called functions in the ROA. By the way, this infographic is posted at the web address shown at the bottom of the slide. The 4 core functions of the collaboratory are indicated by rectangles in the bottom half of the figure. In the research strategy section of the application, applicants are allowed up to 5 pages to provide their plans for each function. Section 8.3 of the ROA has detailed application instructions. Refer to Section 1 of the ROA for function descriptions and a list of key tasks under each function. Applicants may provide alternative tasks to those listed, but if you do, you must indicate how your alternatives would replace the listed tasks. So function 1, indicated in the bottom middle with the green outline, is the project administration function, also called the central coordination core, which will be led by the prime recipient. This function will provide general oversight of the collaboratory, keep track of budget spending and project milestones, manage meetings and communications, receive variant calls from the sequence and analysis function core, use those derived data to assess whether participating newborns are screened positive or negative, and provide general coordination across the study, including coordinating among the 5 to 10 participants in the state public health lab. Participating in state public health labs. Next, there is a community advisory board and LC function, which is depicted with the blue-purple outline on the bottom left for the community advisory board, as well as the gray-blue outline of the entire collaboratory for LC. The community advisory board, or CAB or CAB, must be formed within 3 months of the award being issued and should include the perspectives of parents and guardians of newborns, community leaders, healthcare providers, genetic counselors, newborn screening advocates and professionals, bioethicists, and data privacy experts. The CAB will provide guidance on participant recruitment, informed consent, the target gene list, return of results process, privacy safeguards, and the data management and sharing plan. This function will also be responsible for an LC research study that will examine the impact of newborn screening by whole genome sequencing on newborns and families, state newborn screening programs, and/or public perceptions of newborn screening. The participant recruitment, consent, and return of results function at the center in light blue outline will develop a participant recruitment plan in communication with the state PHLs, develop the informed consent process with input from the CAB, communicate with state PHLs about the transfer of the dried blood spot punches from participating newborns to the collaboratory, oversee the return of screen positive results to participating families, and guide participating PHLs on referral and clinical workup recommendations for the screened diseases. And lastly, the sequencing and analysis function, lower right, will determine the target gene list with input from the CAB, and will also run the genome sequencing pipeline for all consented samples, securely store the genomic data during the study so the data can be reinterrogated if the target gene list is optimized during the study, and transfer the derived variant calls to the administrative function or the central coordination core. Next slide, please. The overall purpose is to support a milestone-driven study with significant community involvement that will assess the feasibility of a collaborative model for incorporating whole genome sequencing into the existing state-based U.S. public health newborn screening program. During the award negotiations, the potential awardee, together with the NIH working group, will select 5 to 10 state PHLs from a PHL roster that the NIH working group is developing. The CAB will provide input to guide development of the informed consent process, list of genes to screen, return of results process, and genomic data sharing plan as previously discussed. Please note that this CAB must be formed as soon as possible post-award or within the first 3 months of the award in order to provide input on these processes. Next slide, please. The anticipated work product for this collaboratory includes screening of participating newborns by whole genome sequencing in 5 to 10 states that have different levels of experience with NIH-funded research and/or with whole genome sequencing, to conduct an embedded LC research study, as well as to provide a final report. If successful, this study will demonstrate the feasibility of adding whole genome sequencing to an existing public health resource available to all babies born in the U.S. The initiative will provide federal and state partners with critical data needed to inform sustained implementation of NBS by WGS. Next slide, please. Some of you may not be familiar with other transactions, or OTs. OTs are not grants, cooperative agreements, or contracts. They allow flexibility to adjust the range of activities, milestones, and deliverables based on the objectives and needs of the program. This means that tasks, partners, expertise, and so forth can be added or removed based on changing research landscape or to address unexpected roadblocks. Many NIH policies apply to OTs, though not the traditional policies governing grants or contracts. NIH proposals for OTs are required to go through an objective review, which is a scientific evaluation managed by an OT meeting administrator. The OT team must develop the criteria and composition of the review team designed to ensure integrity, fairness, and transparency. Award funding is different from typical NIH grants. For example, OTs allow for variable segment lengths, no future funding commitments, and early terminations or extensions by NIH depending on the circumstances. Changes in the project and budget are also handled differently. For example, NIH can propose changes beyond what was required in the ROA, as well as establish unique terms and conditions, like a threshold for prior approval that's customized for each individual award. Next slide, please. Applicants are required to submit a draft data management and sharing plan with their application that outlines and justifies any potential limitations needed to protect the privacy and confidentiality of newborn participants. The final DMSP will be developed in consultation with the CAB and the NIH working group. Next slide, please. More than one individual may be named as a principal investigator, but one must be identified as the contact PI. If a multiple principal investigator application is submitted, then an MPI leadership plan is required. Please note in the ROA and on this slide the percent effort required for the PI, the MPI, and each function director. Next slide, please. A letter of intent is required to be eligible to submit a full application. NIH will use LOIs to plan the review panel and mitigate potential COIs from prospective reviewers. Please note that LOIs are limited to 4 pages and must contain the information in the bullets on this slide. LOIs are due Friday, April 4th, 2025, and must be emailed as a PDF attachment to the email address that is listed on this slide. Next slide, please. This timeline overview shows the key events and due dates leading up to the earliest possible start date for the award project. One thing I want to highlight is that next Monday, March 31st, we are holding a webinar that's intended for public health laboratories. If you fall into that category and want to attend that webinar tailored to the public health laboratories, there's a registration link on the Common Fund website for this initiative, which is provided at the bottom of this slide. Please email us if you have trouble finding the registration link. Next slide, please. Full proposals are due Wednesday, May 14 by 5 p.m. local time of the applicant organization. Please note that late applications will not be accepted. Applications will consist of multiple components, and these components are listed in the ROA along with the page limits for the components and all other application requirements. Next slide, please. Thank you for your interest in this funding opportunity. Additional information can be found on our frequently asked questions website, located at the address shown on the slide. You can also contact our team using the email address that is provided on this slide. Next slide, please. Now we invite you to ask questions about the funding opportunity. You can use the Q&A function in Teams, or you can raise your hand, and we'll let you know when to come off mute to ask your question. We will wait for some questions to come in. So we've received our, oh, go ahead, Matt.
No. I just wanted to make sure you saw that we have some questions in the Q&A.
Thank you. So the first question, can an application be submitted by an institution even if their state PHL is not planning to participate? And that's a great question. And the answer is yes. And it is actually the NIH working group that is developing the roster of those state PHLs, from which the prime recipient will, together with the working group, make the final selection. So it will not necessarily be a state in which that institution lives. So the states for the PHLs does not impact the state from where the prime recipient is. Matt, would you like to answer the review question?
Sure, I'll take a shot at answering this one. So the review process is very similar to the way that grant applications are reviewed, except for the fact that our team assembles the review panel and manages the conflicts of interest. And we actually have a person who kind of fulfills the role of a scientific review officer to make sure all the rules are being followed and that the review is objective. The review panel consists of experts in predefined areas, such as experts in data coordination, pediatric diseases. We have a review area for newborn screening and so forth. And so the reason we ask for those letters of intent is that we want to make sure to manage every possible conflict that might arise between one of the reviewers that we selected a priori and an incoming application. So it's very analogous to, the process is called objective review, and we basically follow an analogous process to the standard review for NIH grants, only it's not a grant application, so we have to basically assemble and manage our own review panel. None of the program team will be on the review panel.
Thank you. There's a question about, do we get any feedback ranking from the LOI or the letter of intent? Matt, would you like to?
Yeah, I'll take that one too. There is the letter of intent is only to manage conflicts of interest. It does not have any bearing on the scoring of your application, although we do, those conflicts of interest, potential conflicts of interest are very serious in order to have an objective review. So we do require the letter of intent, and we require you to demonstrate that you submitted a letter by attaching our reply acknowledging receipt in your application, but it has no bearing on the scoring of your application.
Thank you. There's a question of, is there an expectation to show prior work with state PHLs? There is not an expectation or requirement that you specifically show prior work, but if you have worked with state PHLs, you're invited to include that information as part of your application. And then there's a question, can the consent be obtained after the blood spot is drawn? Yes, that the applicant could propose that the consent be obtained after the blood spot is drawn, and just address this and how that consenting process would look in the application. There's a question, can you confirm the minimum number of states required or preferred? Is there a preference if participating states are located in the same region or close to each other? So one of the goals is to really have states that differ in different aspects of this. So they may be states that have not previously worked with NIH research. They may be states that have no experience with genomic sequencing. And so that's why the NIH working group is working to develop this roster from which states can be selected. We are hoping to have a, geographic distribution. So the answer would be no, we don't want them to be states that are necessarily co-located, or very close to each other, because we really want this to be a somewhat representative sample. And apologies, I'm trying to read while answering.
Here's a question, again, on the conflicts of interest, which I will answer. The question is, since the public health labs will be assigned after grant submission, how will conflicts of interest be identified? Well, we are creating, meaning the NIH working group for this initiative, is creating the list of interested public health labs that can potentially participate in the project and have expressed an interest and kind of meet the characteristics that we're trying to cover in the inclusion. And NIH can manage and ensure there's no conflicts of interest in the reviewers regarding the list of public health labs. So the other conflicts of interest would be between named key personnel on your application and our reviewers. And so we will use one of the requirements on your letter of intent is to list the key personnel and their roles in the project and some of their background history in terms of getting NIH awards, experience with IRB processes and so forth. And we will manage those by ensuring that conflicted reviewers are not reviewing your application and are not in the room during discussions.
Thank you. There's a question, is there a particular workflow or method designed to communicate consented patients with the PHLs? And really that is up to the applicant to propose and design, and may need changes during negotiations depending on the specific needs of particular PHLs that are selected, since the applicant is not designing this with a specific set of PHLs already selected. There is a question about I apologize, I'm losing it. Does WGS mean Illumina, short read technology? And we are platform agnostic. And can NGS approaches other than whole genome sequencing be proposed? And really our intent is to use whole genome sequencing as the platform, which allow the technology really that's used to get the sequencing. As mentioned, there will be an expert panel that selects a list of conditions that will be screened for using this and resulted. But the idea is that the data is there should that list change during the time of the study, which is why whole genome sequencing has been proposed. If you have more specific questions, please feel free to reach out to us so that we can answer those.
Dominic, can I identify a couple questions here?
Yes.
Yeah, apologies, everyone. We were used to using Zoom, and now we have to use Teams. So we're not as familiar with the Teams platform for doing these kinds of webinars. But I think a few questions got skipped here, important ones. And I'll let you decide if you want to answer, Dominique, or call on someone else from the working group. Here's an important question, and I hope you didn't already say this. I don't think you did. What is the size of this effort? For example, number of participants enrolled, number of, I guess that means participants whose genomes will be sequenced. What is the intent on that?
We encourage applicants to work within the budget, and it is up to the applicants to propose what number is feasible within their budgetary constraints and everything else, the other aspects of the program. So we do not have a set number.
Okay, very good. Here's one. I'm not sure if we skipped this intentionally or not, but I'll answer it. Given spending cuts in the NIH, how guaranteed are the funds for 3 years? Well, I would say that we're as confident as we can be that we would like to aim for a 3-year initiative here, and all fiscal planning has taken that into account. Although continuation of a funding and award will be contingent on meeting milestones and showing sufficient progress. We would like this to be a 3-year initiative, and we're as confident as we can be in the current climate that we will achieve a 3-year initiative. And I think, has this question been answered? Without knowing what states this will operate in, how can we propose a set of sites, for example, hospitals, who will recruit and consent participants and collect their blood samples? Are we expected to have clinical partners lined up in all 50 states?
So the answer to that is no, of course, because that would be we're not expecting you to have clinical partners lined up in 50 states, but to propose the framework. And, this will be, thinking of incorporating it with the screening programs, that already exist, and being thoughtful about that consenting part of it. I see what you're saying. How are you going to specifically recruit and consent those participants? And so you'll have to be broad in your application. And once those states are identified during the negotiation phase, then working with the NIH working group, these details can be further fleshed out.
Dominic, could you check the questions and see if you can identify ones that haven't been answered yet?
Yes. There's a question, since PHLs will be assigned after grant submission, how will conflicts of interest be identified?
Yeah, I answered that one already.
I'm
We
sorry.
can dismiss that one. Yeah.
A question on should we try to anticipate budgets for working with PHLs or develop those after? We would advise to generate, to put forward budgets, knowing that they will have to be finalized once we know, once the potential awardee is in negotiation and we have the PHLs identified. So put in just an anticipated budget. Now, did we answer the one about small business or for-profit organization? I can read it out. I don't think this was answered. Regarding eligible entities able to apply, you list small businesses and for-profit organizations other than small businesses. Do you mind elaborating what constitutes as a small business and a for-profit org? That one
I'm not a small business expert. There is a definition, I think, but basically all small and large for-profit entities are eligible to apply if they're domestic. Does anyone else have? We can follow up to the person who asked that question. Was that an anonymous question, or we will post something?
It was anonymous. We can put in the FAQ.
We'll put an FAQ on it, but I'm not sure what the distinction is. But basically, domestic for-profit and non-profit entities are eligible to be the prime applicant and the prime recipient.
Thanks. So another one is the intent to establish or to scale the implementation. And really this is implementation of this type of genomic sequencing into newborn screening will be a much larger project. So this is really looking at the feasibility of this collaborative state model using genomic sequencing, and to pressure test some of the implementation aspects of this that will need to be addressed. So this is not answering all of the questions, and therefore this is not an implementation project, but rather looking at a subset of the questions that need to be answered. And as mentioned earlier, this provides some of the data that other federal partners will be using in implementation strategy in the future if this demonstrates feasibility. Matt, a question, is a budget required as part of the LOI?
No, the budget estimates are not required as part of the LOI. And I think other members of our working group are monitoring some of the questions, and this interface is a little hard to track. So we invite other members of our working group to come off, turn on their cameras and come off mute if there's a question that we that we didn't get to help us answer. But we can still keep looking at the Q&A here.
There's a question, do we need to be registered in ERA Commons prior to submitting an LOI? And the LOI is something, or Matt, did you want to answer that?
No, please, go ahead.
I say the LOI is emailed to us as a PDF. You will need that ERA Commons login. And so it would be advisable to start the process, but it doesn't need to be completed at the time of prior to submitting that LOI.
Correct. You should register, get all the needed registrations started for submitting an application. Basically start that process now if you haven't done so already.
All right. Matt, this question, as there will only be one award for this ROA, is there any sense on whether there will be another reissued, or is there a possibility of other NIH ICs getting involved in funding this beyond the 3 years of the current ROA?
Well, those questions asking us to predict the future are always a lot of fun. So we hope you can clearly read between the lines and see that our hope here is to facilitate and inform an expansion of this. But we cannot yet say if there will be a follow-up funding opportunity. Generally, as a general rule, we can't announce funding opportunities before they are put together and published. But we are pointing in the direction of a continued effort, but we don't have follow-up funding opportunities prepared as of now.
Thanks.
Here's a very kind of procedural question. Is there an expectation of a minimum sample volume? Dominique, would you like to take that, or?
I believe I may have answered that.
Oh, did you? I'm sorry.
But if not, we just advise applicants to consider what they think would be an appropriate number screened and what fits within the budgets in the context of the entire program.
And regarding the samples for whole genome sequencing, those are going to be dried blood spot punches, and one of the sequencing. One of the requirements for the sequencing function is to kind of do a quality control to ensure that the dried blood spot punches are going to give. And Melissa just came off mute. Melissa, do you want to answer that question?
Yeah, I was just going to say that the sample required is punches from a dried blood spot card. And so the proposal should include what they think is the minimum number of punches required in order to adequately perform the whole genome sequencing.
Thanks, Melissa. Another question, given whole genome sequencing is expensive, is it the intention to focus on low-cost approaches to whole genome sequencing? And the answer, I believe I'm going to be answering this, the question that was asked, hopefully, but no. But we are using whole genome sequencing platform, and we do anticipate that the cost will continue to go down in the future, which will help with future implementation of this type of sequencing for newborn screening, but the intention is not necessarily in this ROA to focus on low-cost alternatives or approaches.
And here's a question that asks for clarification about budgets in the letter of intent, which I'll respond to. So the question here is, under the LOI section of the ROA, it states that we need to list all key personnel for the proposed project and for any sub-projects with proposed budget. So it seems that budgets are needed. Can you clarify, since I just mentioned that budgets are not required? So we don't need budget tables and budget estimates, but I suppose, yes, you need to have enough of a scope of what you anticipate is going to be your application so that you're going to be able to list key personnel on anything that you intend to ask for budgetary support for. So to me, that doesn't count as budgets, but yes, you need to understand what you intend to apply for and list all the personnel on components where you will be requesting budget, but you don't need to provide the actual budget requests in the anticipated budget requests in your letter. I hope that clarifies it.
I'm going to address two questions regarding the public health labs. So one is, can you confirm that the quote bidder does not have to submit the states which it intends to partner with or PHL partners, and those will be decided later? So yes, to confirm, the NIH working group is currently developing a roster of state labs. We intend to have more than 10 state labs on that list, and the potential awardee will then, at the time of negotiation, once awarded, will be able to, or at the time of negotiation, be able to see that list, that roster list, and select 5 to 10 for their study. So I hope that answers that question. And another question is, our institution works closely with our Department of Public Health to implement newborn screening, especially follow-up. How do we express interest or apply to be one of the 5 to 10 states? We have sent out information through APHL to the state labs, so with how they can contact us to share their interest to be on the roster. It is not a commitment yet, but at least their interest. So the state lab can respond to that, as well as we mentioned that we're having a webinar next Monday for specifically focused on the public health labs. So that would be another opportunity to get more information. And if neither of those are sufficient, please feel free to email us for specific questions. And another is a PHL that screens for, oh, yes.
So I'm just curious. So any state public health lab representative can also email us if they're interested in being on the list, and we can start a discussion with them.
Yes.
Is that right? Okay.
Yes. Thank you for clarifying. There was a question, another PHL related question. Is a PHL that screens from multiple states considered a multiple state collaboration? Yes. We would not anticipate that one PHL would be in the final program, even if it does screen for it serves as the regional lab for multiple states. But yes, it is considered a multiple state collaboration.
Apologies if we're not doing these questions in order. The Teams interface for this is a little hard to follow, but here's a very interesting question that I'd like to read. Can a function described in the ROA be performed by more than one team? For example, the ROA states that the prime awardee must lead the central coordination core, but certain tasks within that function, such as community of practice, Can certain tasks within that function, such as community of practice, be performed through a subaward? Well, OTs have a lot of flexibility built in, and I would say yes, certain tasks can be performed through a subaward, and yes, a function described in the role can be performed by more than one team, but there needs to be a clear and convincing leadership structure and plan so that the central coordination core is kind of administering the whole project. But there's flexibility, and our reviewers will be, if you submit a convincing proposal with plans that kind of deviate from a one-to-one correspondence between functions and teams, and you have the right expertise and the right environment, and it's all being pulled together by a strong central core, then I think there's a shot that that would be reviewed as meritorious. So I would say yes. Does anyone else on the working group feel otherwise from my response? Okay, good.
Yeah. I'm going to answer two questions here. The first one, only dried blood spot or any other sample type is accepted. Because we are looking at the feasibility of incorporating genomic sequencing into the current newborn screening program, which does use dried blood spot, the answer is that only dried blood spots will be accepted because it is not doing this study. This research study will not otherwise collect samples from newborns. It is dried blood spots that have already been collected for the existing newborn screening program. So I hope that that makes that clear. And a question of if CLIA labs are required, and this would need to comply with all federal regulations related to returning positive results of participants. So for confirmatory diagnostic testing, you would need to follow the regular procedures, which would include CLIA labs.
Here's a question I'd like to address. Again, I might be going in reverse order here, but apologies if that's the case. So the question is, if feasibility is the goal, how is cost considered in the evaluation? Would lower cost testing approaches be considered positively? It's possible, and I would say likely, that lower cost testing could be considered positively because, we wish that we had more funds earmarked for this because it could be a much larger feasibility study, 20 states, public health labs, if possible, but we have budgetary limitations to work with. And so lower cost testing would allow potentially more participants to be included in the study, and because you have to be within the $4.8 million budget limit. And so lower cost testing, if it's a real feasible approach and a solid approach, allows more participants, and that could very likely lead to a more favorable review, in my opinion.
Thanks. Question. So Revity served states would not be on the list of PHLs, or would it just be that Revity would not be on the list of PHLs? So the response is that only the state public health labs are on the public health lab roster, irrespective of who serves those states currently. So the PHL roster includes only the public health labs. And then a question regarding confirmatory testing on positives. What are acceptable molecular testing? And this would be for the applicants to propose what they would plan to do for confirmatory testing. Another question on the budget for PHL involvement. So how do we budget for PHL involvement and expense if NH is curating the final list? Man, did you address this? I don't think you did yet. Right. Really, the response is that we understand that it'll be an anticipated budget because you won't know the final budget until you have the exact PHLs that you're working with. And so this is an anticipated budget. And as you'll see in the row of the budget does get finalized during the negotiation period.
Yeah, that is correct. I'll just add that we have some guiding text in section 8.5 under budget details. And the essence of it is, do the best you can. And during award negotiations, that is a process, that is a stage when we will work things out in negotiation with the prospective awardee. And if you can provide any projections and estimates of budgets to the best of your ability, please provide that in the application. And refer to section 8.3. I'm sorry, 8.5. And yes, it's going to be far from perfect, but do the best you can.
Another question, can you confirm you're proposing to use only leftover dried blood spots from existing newborn screening cards without recommending any additional cards for this project? And that is correct.
We answer I know you read it out, but I'm trying to moderate a few things here at the same time. The confirmatory testing and acceptable molecular testing for confirmatory testing, do we want one of our other working group members to weigh in on that? Or did you address that, Dominique? Apologies if you did.
I did address, but if anyone else from the working group wants to, come on.
Yeah, so CLIA is obviously tricky, especially in the research setting. CMS would say that at any time you're reporting out an individual level result, you need to be doing it from a CLIA certified lab. And so we anticipate that the whole genome sequencing done would probably need to be done from a CLIA certified lab or CLEP if you're working in New York State. And certainly the modality of confirmatory testing will depend on the exact genes that get selected for the virtual gene panel, but we expect that would also be CLIA compliant.
And to also clarify that we anticipate that we would be returning positive results only, and estimated to be about 2% of the population screened.
Thank you both. I'm going to clarify. So the question, can you confirm you're proposing to use only leftover dried blood spots from existing newborn screening cards without recommending any additional cards for this project? We are enrolling new participants, so we are not, what I meant is we're not collecting additional cards on any newborn, but we are not using stored NBS cards from newborns born in the past. So this is a prospective study. So I hope that clarifies that. There's a question, could you comment on disease priority? Is it existing newborn screening disorders or those that can be in the future? We will have, as you'll see in the ROA, that there will be an expert panel that is brought together to determine the final list of conditions that are screened for. And this is These individuals will obviously still get the newborn screening that their state health lab runs. This is not to overlap with those conditions. So this is other conditions that are not currently screened for. And certainly those that are not on the RUSP, many of which have good biochemical assays for screening. Melissa, there was a clarifying question saying based on what Melissa's statement that we're only returning positive results. The expectation is that negative results would not be returned to participants. And that is our expectation. But we could always discuss further offline if there are further thoughts on that. Another question on the blood spot. Can we collect an extra blood spot at the time of clinical NBS? Our state is very protective of their spots, and it could be a problem with participating. We'll work out those details with states during negotiation. So I'm glad that you bring this up as a potential concern, and we'll certainly work with states as we're generating the roster and discussing with them.
I think there's one question remaining. We can make it the last question. And of course, anyone can reach out to us at our mailbox, which was given in the presentation after the fact. But I think we can make this the last question, and it's a little bit of a tricky question. So the question is, sorry, still confused about how recruitment and consenting will work, given that we won't know which states this will operate in. Suppose our clinical team is based in Massachusetts, but all of the chosen PHLs are from other states. Would our Massachusetts-based clinical team be expected to perform recruiting and consenting of patients located in the states where the PHLs reside?
So they need to propose a consent process that could be implemented in other states and has the ability to reach parents statewide, as we again are looking to assess the feasibility of this as a program that could be implemented statewide across multiple states to ultimately understand how it could be and what the barriers are to incorporate it into newborn screening, which of course is statewide.
And I'll add to that we're trying to cast a very broad net on including PHLs on the list. If we had the outcome we're hoping for, we would have all PHLs on the list. And so we're going to try to get the Massachusetts PHL on the list. We are at the top of the hour. Thanks so much for attending our webinar. Please feel free to reach out to us. Is this a good stopping point? Is the working group in the process of answering questions? I think we have to stop here to keep on schedule.
We could follow up on the remaining questions. No, just thank you very much for attending this webinar and for all these excellent questions. Matt, I will hand it back to you.
Okay, thank you, everyone. And again, we have the PHL webinar, which is for PHL reps only a week from today. Information is on the website, and we look forward to having a discussion with the PHLs next week. Thanks again. We're really excited about this webinar, and we look forward to seeing your letters of intent and eventually your applications. Have a good day, everyone.